Aloha. Last time we finished our discussion of RNA transcription in prokaryotes. Today we will focus on RNA transcription in eukaryotic systems or eukaryotic cells. To begin, let's introduce the players, or who does the synthesis of RNA in eukaryotic cells. The RNA polymerases, as you can see on the slide, there's RNA polymerase 1, which synthesizes primarily pre-rRNAs. In other words, this polymerase is responsible for the synthesis of the RNA that's going to be part of the ribosomes. RNA polymerase 2, or POL2, is the polymerase that synthesizes the messenger RNA and some other regulatory RNAs that are part of the original RNA or gene structure that is part of the gene. RNA polymerase 3 synthesizes primarily tRNAs and some other regulatory RNAs. The promoter for POL2, or for RNA polymerase in eukaryotic cells, is designated by a sequence. Well, one of the consensus sequences is the Tata box, or the sequence TATAAA, which is found usually about 30 base pairs upstream from the beginning of transcription. Now, there's other sites, such as the INR site, which is almost immediately in front of the beginning of transcription site. But the interesting thing about these sequences is that they are not found in all genes. Some genes have promoters that do not have the Tata box and the INR sequences. In other words, we still don't quite understand everything about the promoter sites in eukaryotic cells, but at least many cells, or many genes, I should say, in their promoters contain the INR and the Tata box consensus sequences as part of their promoter. Right here, as you can see on the diagram, it says various regulatory sequences. It's shown on this graph as this small box equal to the Tata box, but this can be hundreds, if not even thousands of base pairs, and they can be hundreds and thousands of base pairs away from the actual promoter site. So this is a very complex process with very complex regulation. Structurally, POL2 is a large 12 subunit complex. Something interesting about its structure is that it has a large C-terminal domain. From here on, we're going to call it CTD. And that domain, in humans specifically, has 52 repeats of this sequence, YSPT-SPS. Now, what do you notice about this sequence? If you look carefully, you will see that 5 of the 7 amino acids are polar amino acids containing a hydroxyl side chain: tyrosine, serine, threonine, serine, and another serine. Why is that important? Well, the answer will be given with the question, what happens in these amino acids and what is their biological role? Well, if you think about it, amino acids that have hydroxyl side chains are often sites of phosphorylation. And we will see that that is perhaps the reason why we have so many of these repeats and these amino acids in the C-terminal domain, because the regulation of RNA polymerase and the regulation primarily happens by phosphorylation in the C-terminal domain. In addition to that, RNA polymerase 2 interacts with many other proteins called transcription factors, or TF2, or transcription factors, which are required for proper transcription. Now, let's look at the pathway, or the different stages of eukaryotic transcription. The beginning of eukaryotic transcription needs to be with the assembly of the transcription complex. You can see the Tata box, the INR. Will assume that all genes have that, and so we'll use that as an example. First, we have the Tata box, and the transcription factor 2A binding to the Tata box. Transcription factor 2B binds the Tata box binding protein and also DNA on either side of the Tata box binding protein. Transcription factor 2A stabilizes this interaction, but it is not required at all times. As you can see later on, we have transcription factor A, E, and H also associating with the transcription complex. Now we have the complex complete, and I'm sure there are other factors that are not mentioned here, but at least at this point, for our intents and purposes, we will say that the complex is complete. Transcription factor 2H has a kinase and a helicase activity. It's important because that activity will be used for the formation of the transcription bubble or transcription window, whatever you want to call it. And also with its kinase activity and with the help of cyclin-dependent kinases such as CDK9, the C-terminal domain of the RNA polymerase will be phosphorylated. The phosphorylation of the C-terminal domain initiates the transcription process. The RNA polymerase, with some of the factors, leaving some behind, such as Tata binding protein and B and A, begins the transcription of the RNA transcript. After approximately 60 to 70 nucleotides, transcription factors E and H fall off, and elongation factors join in. They stabilize the elongation process until the termination is reached. And when the termination is reached, RNA gets released, the transcription complex falls apart, and the transcript is then transported for processing, and the transcription machinery is ready to begin another cycle of transcription. The next slide I would normally show you is the slide that I normally use to describe everything that I just said so far. From our last experience, I realized that you can see the video, but you can't hear the audio. From my experience, based on my experience at least. So please go to dnalc.org and watch this video. I'm going to start it simply so that I can show you what it is about, but please watch it later on. Okay, so here you can see, here the DNA molecule that is added, that has some transcription factors bound to it. And at some point you will see how the transcription complex will assemble. Well, it's quite funny because I can hear the audio, but I know you guys can't. And I don't want to simply transcribe or repeat what the narrator is saying. You can watch the video later, but here it is. The transcription complex is formed, but notice it's not starting yet. In order for it to start, you need to have that activation, or an activation factor needs to bind. That activation factor can be thousands bases away. The activation protein or activation factor is bound to an enhancer segment of DNA. When that activation protein binds to the initiation complex, of transcription initiation complex, transcription will start. And as you can see, it took off that yellow ribbon that is coming out of the transcription complex is the newly synthesized mRNA. You can see how the two strands of the DNA are split temporarily, so that the template strand can be used as a template. All these yellow butterfly looking things that you see are the nucleotides, the ribonucleotides that are used by the RNA polymerase to synthesize the mRNA. Once the mRNA is fully synthesized, it gets released, and it's going to be taken out for processing. All right, again, go to dnalc.org and watch this video on their website. I quite enjoy these videos. They're very informative. All right, to continue on, we have transcription, obviously, but as you know, and you've heard me say before, if there is a process, there must be a regulation. So how is transcription regulated? Primarily, this regulation occurs through phosphorylation and protein interactions. Of course, transcription is a target process for drug development, and you can see many antibiotics have been synthesized to help us fight infections, such as actinomycin D, acridin, rifampicin. There are some natural toxins such as alpha-amanitin that are inhibitors of transcription, and as such they act as poisons. It would be interesting to mention, since there's an outbreak going on, many people would say, well, why can't we generate drugs against viruses? The only problem with developing drugs against viruses is that, well, viruses use our own machinery, the host cell's machinery, to propagate themselves. In order for us to attack the virus, we need to attack that machinery. We cannot really do that because that's basically taking a poison for ourselves. So that's why the fight with viruses is not so much with chemotherapeutics, but with approaches to boost our immune system so that our immune system can fight the viruses and kill the viruses. Okay, let's continue on. One way for a regulation of transcription is by processing the product, or mRNA processing. As you can see, the initial product of transcription is called a primary transcript, or immature RNA, which later on, after processing, becomes mature, or final mRNA. We can call it, I guess, a final. That processing involves three main processes: adding of a 5' cap, which is part of the 5' untranslated region. Also adding a 3' polyadenylation tail, which is part of the 3' untranslated region. And also splicing of the primary transcript into its mature form. Excuse me. Now let's take a look at these processes in more detail. For each of the discussed process, we will talk about what is the purpose, when does it happen, and how and where it happens, and how and where it happens. Okay, so 5' cap. What is the purpose of the 5' capping? It protects from degradation by ribonucleases and also acts as a recognition site for cap binding proteins during translation initiation. So that is the purpose of the 5' capping. When does it happen? It happens early in transcription, after perhaps the first 20-30 nucleotides, as you can see it shown on this figure. And then who does the capping? I mean, as you can see, there is the cap binding complex that is part of the C-terminal domain, that associates with the C-terminal domain of RNA polymerase, which associates with the cap. The capping enzyme adds a 7-methylguanosine nucleotide that is bound to the first base via triphosphate linkage. In addition to that, as part of the cap, we have methylation on the 2' ends, either of the first or both first and the second nucleotides. Okay, I'm going to stop here because I'm almost out of time, and I will continue in a minute. Aloha. The next RNA processing mechanism we're going to discuss is the addition of a poly-A tail. The purpose of polyadenylation is Well, it could be either to prevent mRNA degradation or to promote mRNA degradation. In other words, we can basically say that the 3' end has a regulatory function that controls the degradation or the half-life of the mRNA. Polyadenylation occurs, well, at the end of transcription. Obviously, we have to have the end transcribed before the poly A tail is added. How does the cell know when to stop transcription? It's been found that approximately 10 to 30 nucleotides upstream of the cutting site, there is a sequence, AAU, AAA, and also about 20 to 40 nucleotides upstream, there is a GU-rich sequence that is not well defined. But these sequences, especially the AAU, AAA, served as a recognition site for an enzyme complex to associate with it and then cleave the primary transcript, removing it from the transcription complex. After this happens, the endonuclease is removed and a polyadenylate polymerase is added, and that polymerase now begins to add only A nucleotides, and the tail that is added could be short, made out of 60 to 80 or more A nucleotides, or very long, something like 253. I've even seen larger poly A tails. So that is the function, and that's how polyadenylation happens. The third processing mechanism of the primary RNA transcript is RNA splicing. Now in order to understand splicing, we need to understand the meaning of the terms exons and introns. In this figure, you can see how we have a primary transcript with green and yellow areas. The green areas represent exons, which are going to be part of the final mature RNA that's going to code for the expression of a protein. There are certain parts that are removed from the initial transcript and not going to produce, and they're not going to be involved in the production of a protein. These parts of the DNA, of the mRNA, are called introns. So basically, splicing is connecting of the exons and excising and removing of the introns. The introns are often believed to be useless DNA, especially in the past, because they're cut out and thrown away. But recently, we have found that introns are often sites for the expression of regulatory RNAs, which are critical for the expression, for gene expression. There are 4 different classes of introns. We have class 1 and 2, which are self-splicing. You can see here on the slide a diagram explaining how the self-splicing introns remove themselves. That happens with the help of a free G nucleotide. And you can see on this diagram how the 3' hydroxy group of the G nucleotide attacks the phosphorus, or the phosphate, I should say, that connects the end nucleotide of the exon and the first nucleotide of the intron, displacing the intron. Then the hydroxyl group at the 3' end of the proximal, the upstream exon, will attack the phosphate, connecting the last nucleotide of the intron and the first nucleotide of the downstream exon. By doing so, following the same chemistry, the intron is removed, and now we have the 2 exons being linked together. This is class 1. Class 2 is follows pretty much the same logic and chemistry, but the only difference is that we don't these type of introns do not use a free nucleotide, but they use one of their own nucleotides, usually within their own sequence, that does the attacking at this location between the last nucleotide of the upstream exon and the first nucleotide of the intron, and then it forms a lariat structure that later on gets removed. So this is class 2. Class 3, introns are removed via spliceosomes. These are, well, most introns are not self-splicing. Class 3 is the largest and usually most common type of introns. And for their removal, the cell utilizes small nuclear small nuclear RNAs, and also uses small nuclear riboproteins. The small nuclear RNPs use the small nuclear RNAs to recognize certain sequences within these introns and exons, and thus it guides the splicing machinery that later on will remove the intron. So let's take a look in greater detail how this happens. The small nuclear RNAs, well, there are 5 snRNAs that are used in the spliceosome: U1, U2, U4, U5, and U6. U1 and 2 begin the process by recognizing, shown right here, their corresponding sequences. Then they recruit the U4, 6, and 5 to form the inactive spliceosome. With the help of ATP, the spliceosome now is activated. The intron forms a lariat structure and later on gets excised and removed together with the splicing machinery. One thing I forgot to mention for class 1 and class 2 introns is that they are independent of ATP, where these are dependent on ATP. On this diagram describes when the splicing happens. As you can see, it happens while transcription is occurring. So we can have splicing done while transcription is occurring, and we can also have splicing that happens after transcription is done. All right, so that pretty much describes the process of spliceosomal removal of introns. There is another video that I have included here. It's really not that informative, to be honest. It basically describes the same process. So I'm just gonna let you watch this separately. I'm not gonna waste time on the video just to show you pictures, and I'm just gonna move on. The 4th type of intron removal is with the help of endonucleases and ligases. So very similarly to excising of certain DNA with the help of endonucleases, and then a ligase will come in and it will connect the two exons. In this process, we also use ATP. Now, this is the interesting part. RNA processing is not always as simple as we described. In these past slides, it appears that, well, we can add a cap, we can do splicing, we can add a poly A tail, and everything seems to be fine. But remember, I believe in 481, I asked you once, why do we have approximately 23,000 genes while we have in the realm of 100,000 proteins? And then I told you that it's because of alternative splicing. And here, hopefully, you'll be able to appreciate now the meaning of alternative splicing. On this diagram is shown that we have two polyadenylation sites. Based on which polyadenylation site will be detected and used in a given cell, we can have two different products coming from the same gene. On this side, we have it a little bit more complicated. We have one poly A site, but different splicing sites. We have the primary transcript, and we have polyadenylation of the primary transcript. But based on what type of splicing we will have, some of the resulting exons might not be found in the final product, leading to two different products in this particular case. Now let's look at this more specific example. There is one calcitonin gene shown here. This is also the primary transcript. Each green area is the exon, including, well, the green and the blue areas are exons, poly A site, another poly A site, and some introns in yellow. This gene gets polyadenylated differently in different tissues. In the thyroid, we have this polyadenylation. In other words, exons 5 and 6 are excluded because the cell machinery recognizes the first poly A site. In the brain, though, all exons are included because the brain, the first poly A site is not recognized, but instead the second. When splicing occurs, we have two different products. In the thyroid, we have only exons 1, 2, 3, and 4. In the brain, we have all 1, 2, 3, well, except for 1, 2, 3, 5, and 6. When we now translate the products, we get two different products. And when the products are processed, we see that in the thyroid, we get calcitonin, which is basically exon 4. And in the brain, we have a completely different product, which is exon 5. And that product is known as calcitonin gene-related peptide. So the only relationship between these two products is that they come from the same segment of DNA or the same gene, but they're completely unrelated in terms of sequence and most likely in terms of function. So you can see the power here very well demonstrated with a real example, the power of alternative splicing and alternative processing of mRNA. And here's another, this is our final slide, but this slide basically describes the complexity and the variability in mRNA processing and how that leads to multiple and various products. You can see we can have alternative exons. We have alternative 5' splice sites, alternative 3' splice sites, etc., etc., etc. Various products from the same gene, all because of alternative processing of mRNA.